Hello friends, so today we have got the grand test 181 and we are having the session questions in microbiology from 86 to 99. So the first is question number 86. Now this question says, this question is saying all of the following are true except. So now you see the first answer, an epitope is a small portion of a macromolecule. This is correct. Now this epitopes and paratopes, epitope is present on the antigen and paratope is present on the antibody, right? So epitope it's a complex structure. It can be linear, it can be conformational. So this epitope is a small portion of a macromolecule. So it's a bigger molecule on which it is present. The second answer is the variable region domains contain the antigen recognition site. So this is also correct, because if you see the shape of the antibody molecule, so this is a Y-shaped molecule, right? And the variable region, this is known as the FC portion of the constant portion, and this is known as the F antigen binding portion, right? So we have got two FAB and one FC portion, right? So if you see this, the variable region, this part is known as the variable region, and it is here where the antigen binds to this molecule. So the antigen will bind here. So an antibody molecule will be utilizing its variable region for the binding of an antigen. So the variable region domains contain the antigen recognition site. This is also correct. The third option is an IgG antibody is monovalent. Now let me tell you that this is the wrong answer, because no antibody is monovalent. Antibody is always a bivalent molecule, as we saw here that the valency of an antibody molecule is 2, because it is able to bind two antigens at the same time. So the valency is always 2. Now let me give you other examples. So let's see here. So if we have got IgG, the valency will be 2. Now across all the IgG, it's always monovalent. Sorry, it's always a single molecule. So the valency will be 2, right? If you talk about IgA, right? So we have got IgA1, which is present in serum, right? This is present in serum. So again the valency will be 2. But if we talk about IgA2, which is present in secretions, so IgA2 is the antibody which is present in the intestinal secretions, tears, like the colostrum. So the valency will be 4, because this is a dimer. So whenever the normal antibody molecule is there, that's called a monomer, and IgA1 is a monomer, whereas IgA2 is a dimer. So the valency is 4 here, right? Now let's see the IgM. Now IgM is a pentamer. So we have got two types of IgM also. So IgM, which is present in serum, is a pentamer. So pentamer means that the valency will be 10. Whereas we also have got a IgM which is present on surface of B cells. Now this is a monomer, right? So the valency will be 2 again. So because this is so you have to understand the molecular structure. Once you understand that, then you will be able to answer all the questions. Then IgD, the valency will always be 2, because it's a monomer, and IgE is also a monomer, so the valency will be 2. So don't get confused between all these antibody molecules. You just have to have a rehash about the different structures of the antibody molecules, and thus you can conclude on the valency, right? The fourth option, the class of an immunoglobulin is determined by the heavy chain. So this is also correct. Now if you see here, so this is the heavy chain. Now this FC portion, so this portion, and which is primarily comprised of the heavy chain, this determines the type of the antibody. So we have got these 5 types of antibody, like IgG, IgA, IgM, IgD and IgE. All these 5 types of antibodies are determined by the heavy chain, right? So heavy chain is the one, if you see the structure of this antibody molecule, heavy chain is the one which determines the type of the antibody. That's one. Secondly, it has got effector functions, such as this portion will bind to the cells. So if it has to be bind to the B cell, then this portion will go and bind to the cells. and thereafter it can also cause antibody dependent cell mediated cytotoxicity. Now if you see the functions of this portion, or the FAB, F antigen binding portion, or the F variable portion, so this portion has got the function of binding an antigen molecule. Okay, so let's move to the next question. So the question says that a molecular biologist in the lab is analyzing DNA, and so the DNA has got different percentages of A, T, G and C. Now you know that A is complementary to T, adenine is complementary to thymine, and cytosine is complementary to guanine. But normally what happens is that when this molecule is a normal molecule, so if we have got double stranded DNA, so this will be like this. It's a plectonymically coiled structure, and if you go into the ultrastructure, right, then you will see that there are these base pairs. So this is A, G, C, T, A, and complementary to it you will have T, C, G, A, T. So this will always be in the same proportion as, so the A and T component will always be in the same proportion as the C and G component. Now this is normal for a double stranded DNA molecule, but what if it is a single stranded DNA molecule? Then the component of A, T, G and C will vary, and this is because it is a viral genome. So viral genome can be single stranded DNA, and it can have different portions of adenine, thymine not complementing each other, as well as cytosine and guanine not complementing each other. Okay. The next question says, to which cell type does the clonal selection theory apply? So now you see here, clonal selection, this concept is important in the evolution and development of the T and B cells. So when these T and B cells, they both start in the bone marrow, the T cells, they will migrate to the thymus, right, and B cells will be in the bone marrow. But they have to undergo clonal selection. It's a part of the maturation. So this both B and T lymphocytes, they have to undergo clonal selection to become mature T and B lymphocytes, right? This concept is not applicable to neutrophils. Okay, so now the following instrument is sterilized using. So you can see that it is a scope. So all these endoscopes, all endoscopes are sterilized traditionally by 2% glutaraldehyde, which is also known as Sidex. So this is the classical use of glutaraldehyde for sterilization of endoscopes, and this is by these aldehydes, they actually kill by alkylation, right? So they will cause alkylation. Fine. Now in the options we see that we have got formaldehyde. This is not used because it is highly toxic and it is also an irritant to the tissues. And we have got two other, beta BPL gas. This is a gas, beta propionolactone gas. This is used for inactivation of vaccines. So we have got BPL inactivated rabies vaccine. So we use it for inactivation of vaccines. Formaldehyde, this is used for operation theatre sterilization. So in the operation theatre, we generate this formaldehyde gas and keep this OT closed for 48 to 72 hours. In that duration, because of this gas, and this gas, this is a sporicidal gas. So it will kill all the organisms, including the spores, leading to complete sterilization. So this is formaldehyde for operation theatre sterilization, right? Then the third option is ethylene oxide. Now ethylene oxide, this gas is also used for sterilization of equipments. Especially, what are we using here? We are using plastics and rubber. So in the central sterile stores department, there is something known as CSSD, which is attached to every OT. So this CSSD is Central Sterile Stores Department. So all the instruments in the OT which have been used, they are brought to the CSSD for sterilization by various mechanisms, and ethylene oxide is used for plastics and rubber. In addition, the ethylene oxide is also used for heart-lung machine and respirators, right. So the answer here is orthophthaldehyde. It is a superior molecule compared to glutaraldehyde, right. It is used in a concentration of lesser concentration of 0.55%, and this is less toxic, more efficient, but more expensive as well. So nowadays for these scopes, now we are using orthophthaldehyde instead of 2% glutaraldehyde or Sidex. Okay. The next question is not a biological function of complement. Now complement is a very, very important topic. So you should know about these options, right? So the first is direct lysis of foreign cells. So this is correct. Now this is correct because what the complement does, when the complement forms the membrane attack complex, the membrane attack complex is formed, which is C5b6789. Now this membrane attack complex, this causes target cell lysis. So this leads to the formation of pores in the cells, and this causes direct lysis of foreign cells. So this is correct. The second one is opsonization and phagocytosis. This is very, very important because C3b and C4b, they will both act as opsonins, which will lead to opsonization followed by phagocytosis. So what is opsonization? Opsonization is actually the coating of an antigen. So the antigens are coated by this C3b and C4b, and they become more attractive for the macrophages and monocytes, and thus it facilitates phagocytosis. The third one is it plays a role in inflammatory response. This is very, very important. Yes, it does play a role in inflammatory response. So in this also we have got the complement will act. So the functions of complement will be, first is target cell lysis, second is opsonization and phagocytosis, right. Now we are talking about the functions of complement. Third thing it will lead to is the generation of anaphylotoxins, right. Then it has a role in type 2 and type 3 hypersensitivity. Right. So type 2 and type 3 hypersensitivity is very, very important, because in type 2, which is IgM and IgG mediated, we need complement eventually for the effector functions, as well as in type 3 hypersensitivity, whenever there is an immune complex mediated injury, there also complement gets deposited along with this immune complex and thus causes problems. Then the complement fixation test. So we have got so many complement fixation tests. For example, Wassermann complement fixation test for syphilis, right. Then we have got Treponema pallidum immobilization, that is also a type of complement fixation test. So it is important there as well, right. Then it has also got a kinine like activity. Okay. So these are the functions of complement. This you should know, and questions can be asked on this. So here the answer is this, because it does not aid in the clearance of viruses. So the question is saying not a biological function of complement. So the answer is it does not aid in the clearance of viruses by activating CD8 T cells. Now remember that the complement gets activated, the classical pathway of the complement, it gets activated by the antigen and antibody complex, right. So CD8 cells are well before the formation of the antibodies, because, right, so this is the at the T cell level. So this is not a function of complement. Next question, the image given below shows. Now you can see that this is a Lowenstein-Jensen medium. This is a very standard, this is a very, very standard and a simple and a very easy question. So this bottle is known as the McCartney bottle, and this is one media which is always kept in a bottle. Now the question is that why is it so that other media are poured in a petri dish, right? And this media is, why is it so that the other media are poured in a petri dish, but this media is not in a petri dish, and this media is in a bottle? So the reason behind this is that why this media is in a bottle is because of two reasons. One is that Mycobacterium tuberculosis is highly infectious, so the infective dose for Mycobacterium tuberculosis is 1 to 10 bacilli. So that means that even if one bacteria goes inside your body, then you are going to get infection by Mycobacterium tuberculosis. So we don't keep it in open petri dish, we have to keep it closed in a screw capped McCartney bottle. This is one. Second thing is that this is an egg based media, right? And LJ medium is an egg based medium, and this medium is sterilized by inspissation. Now because of the egg content in this media, maggots, they will start growing in this media very fast, and other bacteria also grow. So to prevent contamination of this medium, we have to keep it in a screw cap bottle. The third reason as to why it is in this bottle is because the generation time of Mycobacterium tuberculosis is 15 hours. So as a result of that, the colonies will be seen in 2 to 8 weeks. That's a pretty long time. So in this time, other media in a petri dish may go dry. That's the reason why we are keeping it in a slant, and this has got a thicker slant. So it has also got some fluid, so as to keep it moist. So this media is poured in a slant in a McCartney bottle, so that, and it's a very thick slant, so that it does not dry during the 8 weeks of incubation, right? Then now what you see in this picture, if this is A and this is B, so this is A and this is B. Now you can see and you can also identify the type of growth in this. So Mycobacterium tuberculosis, right? Mycobacterium tuberculosis shows eugonic growth. Eugonic growth means that it grows well, right? And how will it grow? So it will exhibit rough, tough and buff colonies. Rough, tough and buff colony. So now you see in A, in the A part, you can see that this is Mycobacterium tuberculosis because it is growing very well. It's almost covering the entire slant of algae medium, and the colonies are appearing as rough, tough and buff. Whereas if you see this, the second one, that is M. bovis. M. bovis, this has got a dysgonic growth. That means that it does not grow very well, it grows poorly, right? And this will not show this kind of colonies, so the colonies will rather be not rough, tough and buff. So this is Mycobacterium bovis, right? Dorset egg media is another egg media, but it is not this media is not that. Another thing in this media, what you can see is that the color of this is blue-green. So the color of this medium is bluish-green, right? Bluish-green. So this color comes from malachite green. So in LJ medium, we add malachite green, and this makes it selective. That means that in LJ medium, only Mycobacterium tuberculosis and other mycobacteria should be growing. So we add malachite green, which is a dye, and this dye kills other bacteria. So Tinsdale media is used for Corynebacterium. This is another selective media for Corynebacterium diphthery, right? And we have the Thayer-Martin media. This is another selective media for Neisseria meningitidis as well as Neisseria gonorrhoeae, right? So these are we have got 3 selective media here: Lowenstein-Jensen selective media for Mycobacterium, Tinsdale for Corynebacterium, and Thermatin for Neisseria. The next question is drug of choice for strongyloidosis. Now, none of the options are correct here because the drug of choice is ivermectin. Right? Now, ivermectin is also used for onchocerciasis. Right? So, for onchocerciasis as well as for strongyloidosis, we have to use ivermectin as the drug of choice, and sometimes we also use it for myiasis. Myiasis is when maggots are there, right? Then also we have to use ivermectin sometimes. Now, in these options, if we don't have this ivermectin as a choice, then our obvious choice should be albendazole, because the drug of choice for all nematodes is albendazole. Okay, so let's see the drug of choice for all cestodes. Cestodes are tapeworms. Drug of choice for all cestodes is praziquantel, right? Except neurocysticercosis and hydatid cyst. Now, in these two cases we use albendazole, where we use albendazole. Okay. Now, the next thing here is the drug of choice for all trematodes. This is also praziquantel. Trematodes are flukes. This is also praziquantel. Now, with exceptions, so exception here is just one, that is the fasciola hepatica, except fasciola hepatica, which is the liver fluke, for which this we use triclabendazole. Right? Then next thing here is the drug of choice for all nematodes, or they are also known as roundworms, right? So, for all nematodes, the drug of choice is albendazole. And which are included in nematodes? We have got the Ascaris or the roundworm, Trichuris or the whipworm, Trichinella, then hookworms such as Ankylostoma duodenale and Necator americanus. Then we have got all the filarial worms, right, such as Wuchereria bancrofti, Brugia malai, and Loa loa, Onchocerca volvulus, Mansonella. These are the filarial worms, right? Then we have got Enterobius, which is the pinworm, and we also have Onchocerca, which is another filarial worm. So these are the various worms which we have to study in nematodes. So the drug of choice for all nematodes is albendazole. Here also we have got certain exceptions. So the exception is firstly we have got Enterobius. Enterobius is the pinworm. Here we use mebendazole as a drug of choice. Then for strongyloides is another nematode. We use ivermectin. Similarly, for Onchocerca volvulus, which is a filarial worm, we will use ivermectin. For filarial worms such as Wuchereria, bancrofti and Brugia malai, we use diethyl carbamazine, right? So this is how this is quite different for all this. So these are the various exceptions for nematodes in which we are not using albendazole as the first choice. And also for Dracunculus. Dracunculus is the guinea worm. It has been eradicated in the year 2000 from India and Pakistan, right? But this is still in Africa. So for this we use metronidazole. So these are the certain exceptions in the treatment of nematodes, but the drug of choice is albendazole. So here in this question we have to answer albendazole as the choice here, right? Okay. So now we come to the next question, which is saying which is the least common chronic complication of measles. The answer is very obvious. It is SSPE. It is saying least common, right? So it has to be a very rare disease, one. Secondly, it has to be chronic. Now, diarrhea generally is not chronic in viruses. Pneumonia again is not chronic. Measles will cause atypical pneumonia, but it will not be a chronic pneumonia, right? Otitis media is also quite transient. So these are basically acute infections, right? But SSPE is a kind of slow virus. So what do you understand by this slow virus? Slow virus is a virus which replicates very slowly, and we have got two important slow viruses which we are going to discuss now. So first is the measles virus, which causes SSPE or subacute sclerosing panencephalitis, right? So in slow viruses we have two important ones. First is the measles virus. This will lead to subacute sclerosing panencephalopathy, and second one is the JC virus, right? JC virus is this is actually a polyoma virus. So this virus causes PMLE or progressive multifocal leukoencephalopathy. So these are the two viruses which are known as slow viruses, which will cause, and these are very rare, and these disease entities are also very rare. The next question is, which anticoagulant is used when blood is sent for blood culture? Now in this, when we are sending, now you must have been knowing about vacutainers, which have got sodium citrate, EDTA, oxalate, and they have got various functions, right? But there's a big difference here. So blood culture is not done in a vacutainer. Blood culture is done in a bottle. This is known as a blood culture bottle. This blood culture bottle has got a liquid media, and this liquid media is known as the brain heart infusion, right? So this contains brain heart infusion. And so what is there in this? We have got calf brain and beef heart, which is present in this medium, right? This is the blood culture medium. We have got calf brain and beef heart, and this blood culture is incubated for at least 5 days. So this is incubated for 5 days. After 5 days, if there is no growth, then we put it as negative. Otherwise, we have to keep this under incubation for 5 days. Blood culture is generally done for which diseases? So blood culture is a standard thing for salmonella. So for all the typhoid is diagnosed in the first week by blood culture. So in the first week we do blood culture for salmonella, and this is the most definitive test for diagnosing typhoid. Blood culture is the most definitive test. It has got the highest specificity, right? So that is done for salmonella and brucella. Now this blood culture includes this SPS as the anticoagulant. So this SPS is sodium polyanethol sulfonate. You must have seen the discussion. So we have already given there sodium polyanethol sulfonate is there in this blood culture, and this acts as an anticoagulant. So this is an anticoagulant in this blood culture bottle, right? This is also anti-lysozyme, because this lysozyme which is present in the blood of humans, that can inhibit the growth of bacteria. So is complement. So it is also anti-complement, and it also has got a dilutional effect. So because of this, the blood which is taken from as a sample, that is diluted many folds. So the effect of the inhibitors for growth is reduced, and thus this blood culture bottle is good for blood culture. Next question says that memory T cells can be identified using the following markers. So the answer here is CD45RO. So you should know that there are certain important markers, and these markers, these markers, when we are talking about them, then these are known as immunophenotypic markers. phenotypic markers. So we can identify a cell by a marker, and this is an immunophenotypic marker. So which are the important ones? For T cells, the markers are CD3, CD4, CD8, right? For B cells, the markers are CD19, CD20 and CD21. So what is this CD? Now CD stands for cluster of differentiation. CD is cluster of differentiation, right? So these CD markers are existing on lot of cells. So let's say these are the important ones, but you will have macrophages, they will have a different marker. Every cell will have a different marker, right? So that's why when we call CD8 cells, so these are T cytotoxic cells. Cytotoxic cells will have this marker, and this is T helper cell, right? So the CD4 marker is present in the T helper cell, and this marker is present on the C cytotoxic cell. CD8 is present on the T cytotoxic cell. Fine. So now this CD markers can be identified by a technique which is known as flow cytometry. So flow cytometry is a technique by which we identify the immunophenotypic markers on various cells, right? Other than this, we can also use immunofluorescence. So detection of these immunophenotypic markers is by flow cytometry or immunofluorescence. Okay. So the question was asking that memory T cells can be identified by which immunophenotypic marker? So the answer is CD45RO. 96. Now it says most potent stimulator of naive T cells. The answer is mature dendritic cells. So now you should know what are these dendritic cells. So we have got something known as antigen presenting cells. Antigen presenting cells. Now these cells are B cells, dendritic cells and macrophages. So these are known as the antigen presenting cells, right? Now what happens? So if you see the basic concept of antigen processing, right? So in this processing we have most antigens. First concept is that most antigens which are there are T dependent antigens. So most antigens that we have are T dependent antigens. Then the next step in this T dependent antigen is that this antigen goes to the antigen presenting cell. This antigen presenting cell can be of three types. They are B cells, dendritic cell and macrophages. These are the main antigen presenting cells. The third concept here is it goes to this antigen presenting cell takes this to the T cell. Now this T cell is a resting T cell. So it will convert to effector T cells, which are two. So it will form the T helper cell and the T cytotoxic cell, right? So what will happen to these two cells then? So now this T helper cell, this has got the CD4 receptors and this will act by MHC2, and this has got a CD8 receptor and this will act by MHC1, right? So this MHC2, it is for extracellular antigen, right? So if that antigen which we are having initially is an extracellular antigen, that means that this antigen is there in the outside the cells, in the serum, in the blood, then it will be killed by the T helper cell. But if it is intracellular, it is inside the cell, then in this case the antigen is not available for killing by antibody. So what will happen? Then the cell inside which the bacteria or the virus is there will say, hello, I have got an antigen inside, right? So what to do now? So now the entire cell will be killed. So that is killed by T cytotoxic cells. Cytotoxic means that it is toxic to the cell, cytotoxic. So this will be done by the CD8 receptor containing T cytotoxic cell, and this is by MHC1 pathway, and this is for intracellular antigens. And in this process there is a formation of T memory cell. Okay, then this entire setup moves to the B cell. Now B cell is also a resting B cell. So once stimulated, it will form plasma cells. Now these plasma cells, T helper cells, also stimulate the B cells so that it converts to plasma cells. This plasma cell will then secrete antibodies, right? And in this process we will have a B memory cell also. So this is the basics of antigen processing. We have got a T. Most antigens are T dependent antigens. They will be processed by the antigen presenting cell, which can be B cell, dendritic cell or macrophages. Then the system goes to the T cell. T cell can convert to CD4 T helper cell or CD8 T cytotoxic cell, and in the process there will be a T memory cell which is formed. Then the entire system moves to the B cell. The B cell will convert to plasma cell that will produce antibodies, and there will be a B memory cell formation in the process. The second concept is of the super antigen. Super antigen, as the name says, it's a super antigen. So what does it do? So example of the super antigen is staphylococcal toxic shock syndrome toxin, staphylococcal scalded skin syndrome toxin, staphylococcal enterotoxin, and streptococcal pyrogenic or erythrogenic toxin. These are examples of the super antigens. Now because they are super antigens, so they will bypass this step. They will not bind to the antigen presenting cell. They will directly go and bind to the T cell. And where do they bind? They bind to the V beta region of T cell receptor. Right? So we have discussed these two antigens, T dependent, and the second one is super antigen. The third type is the T independent antigen. Now T independent antigen, this will directly go and bind to the B cell, right? Example of this T independent antigen is bacterial capsule, flagella, and lipopolysaccharide. So what is the effect of this? Now once we know that this is there, so the T independent antigens, they will not have a T memory cell. So because of that, once we have got this capsular vaccines, so which are the capsular vaccines? Capsular vaccines are meningococcal vaccine, pneumococcal vaccine, and the typhim VI vaccine, right? These are the capsular vaccines. So this capsule, and also the Haemophilus influenza vaccine, Haemophilus influenza B. So these are the 4 capsular vaccines. So these capsular vaccines cannot, there is no T memory cell in this capsular vaccine, right? Because this antigen is directly going and binding to the B cell. So because of this, the booster and the overall immunity conferred by these vaccines is not very effective. That's the reason why these vaccines are not given in children less than 3-year-old. So these are not given in less than 3-year-old children. So this meningococcal vaccine, right, and these vaccines, sometimes we have to conjugate them in case they are conjugated. So for example, we have got a hip conjugate vaccine. So once we conjugate them, then they become immunogenic. So once we make them into a conjugate, so like we have got a pneumococcal conjugate vaccine, which is the 7, so there are 2 vaccines in pneumococcus. One is a 23-valent vaccine, which is not a conjugate vaccine, that is given only for adults. And the second vaccine is a 7-valent pneumococcal conjugate vaccine. So these conjugate vaccines can be given in children. Right? So isolated capsular vaccines cannot incite the T memory cell formation, and that's why they are not given in children less than 3 years of age. But once these vaccines are conjugated with a protein, then they can be given in children because the immunogenicity increases, right? Okay. So what is this? There are two more concepts here. One is the immunogenicity and the antigenicity. So immunogenicity and antigenicity. Okay, to simplify it, immunogenicity is the ability to cook food, and antigenicity is the ability to eat food. So we all are always antigenic, but we are seldom immunogenic, right? Nobody wants to cook their food, right? So if you see it here, antigenicity is the ability to eat food and immunogenicity is the ability to cook food. So in technical terms, this is the ability to induce immune response. So this is the ability to induce immune response. And this is the ability to combine with products of immune response. Right. So you understand the concept of immunogenicity and antigenicity. Immunogenicity is the ability to cook food and antigenicity is the ability to eat food. You are always antigenic, but we are seldom immunogenic. So immunogenicity is the ability to induce immune response, and capsular antigens are not very good. They are not immunogenic. They are poorly immunogenic, right? Endotoxin is poorly immunogenic. Hapten is poorly immunogenic. And antigenicity is the ability to eat food or the ability to combine with products of immune response. So the T-independent antigens, hapten and endotoxins, they are poorly immunogenic. So they will be, but they are antigenic. So they will be able to combine with the respective antigen-antibody complexes, but they will not induce the formation of the immune response molecules. So once again, see this, there are 3 types of antigens: the T-dependent antigen, the T-independent antigen, and there's a, in between there's a super antigen. So this T-dependent antigen has got these steps: T-dependent antigen, antigen presenting cell, T cell, B cell and antibodies. Super antigen will bypass one step, directly go to the T cell, and T-independent antigen will bypass 2 steps and directly go to the B cell. Okay. The next question is, true about infection caused by organisms shown in this figure is. So now the answer here should be, this is the correct answer, only cyst is infective. Now this does not reside in cecum, this resides in the duodenum, right? For that we do a entero test. So because it is there in the duodenum, so we put a string with a small weight tied and it goes in the duodenum, From the duodenum, we take that sample out and we look for this giardia, right? So what you see here is this is giardia, and this is a trophozoite. Trophozoite means that it is the active feeding form. So once it is happy and it is enjoying life like you, then it becomes a trophozoite, right? So you are trophozoite. Okay, we all are trophozoites. We all enjoy life. And when it has to undergo a stressful phase of life, right? Like just one day before the exam, then we all become cysts. We don't eat food, we don't feel hungry, we don't feel thirsty. So we become like a cyst. So trophozoite is the active feeding form after the exam, and cyst is the dormant and more resilient form before the exam. So the infective stage in parasites, which are the parasites which have to cross the pH barrier in the stomach. So the infective stage of these will be a cyst, because a cyst will be able to withstand the high high acidity and the low pH of the stomach. Whereas the diagnostic stage, when we are trying to diagnose it, then in patients who have got active disease, you will see a trophozoite. So by this duodenal entero test, we will see trophozoite, but in chronic carriers, they will pass cysts only, so you will see a cyst, right? So the answer is only cyst is infective. This is correct. It does not reside in cecum, it resides in duodenum, and we see it by entero test. And this is also wrong, only man-to-man transmission, because this is a orofecal transmission, so it can come from multiple water sources, and it does not exist in one phase, it exists in two phases, the trophozoite and the cyst. Okay, and what you see here, it's a leaf-shaped parasite, and it has got these two nuclei and basal bodies, and there are 4 pairs of flagella. Now these flagella, they keep on rotating, and this is unpurposeful. They don't know what they're doing, but they're just doing it, right? So this will show a falling leaf motility. So falling leaf motility in giardia is because of the flagella which beat in different directions all the time. The next question is, a man presents with fever chills 2 weeks after a louse bite, right? And this is a maculopapular rash on the trunk which spreads peripherally, right? So the cause of this infection B. So now you should understand that there are two concepts here. One is that if the rash comes first on the face, and the trunk, so we call this as the distribution of this rash is and later on this rash moves to the, so initially comes on the face and the trunk, and later on it moves to periphery. So we call it, the distribution of this rash is centripetal, and the spread of this rash is centrifugal, right? So this is seen in chickenpox. So the rash in chickenpox, distribution is centripetal and spread is centrifugal. Now if you see this for smallpox, that's just the opposite. So in smallpox, the rash will first come on the periphery, and then it will go to the center. So here the distribution will be centrifugal, so distribution is centrifugal and the spread is centripetal, and this is seen in smallpox. right? Now the question is also saying that the macular papular rash on the trunk was there. That means it was a centripetal distribution and it spreads peripherally. So what is this disease? So we have got various things. First is we have got fever, we have got a louse bite, right? So which are the louse transmitted diseases? So louse will transmit only 3 diseases. The first one is the epidemic typhus. Epidemic typhus. This is caused by Rickettsia prowazekii. The second one is epidemic relapsing fever. This is caused by Borrelia recurrentis, and the third one is trench fever, and this is caused by Bartonella henseli, right? So we have got, if you see here, louse will transmit these diseases, the epidemic typhus by Rickettsia prowazekii, epidemic relapsing fever by Borrelia recurrentis, right? And trench fever by Bartonella henseli, right? So this is Bartonella quintana. So trench fever is by Bartonella quintana, right? So we have got only these 3 diseases are there which are by louse: epidemic typhus, epidemic relapsing fever, and trench fever. So let's go to the question again and see. Rickettsial pox is by mite, so this is not there. Scrub typhus is also by mite, also not there. Endemic typhus is by flea, so this is also not there. The only answer is epidemic typhus. Plus you should also know that the rash distribution in epidemic typhus will be centripetal and the spread will be centrifugal. Okay, so we come to the last question of the session. So the 60-year-old man is diagnosed with suffering from Legionnaire's disease after returns from attending a convention. He could have acquired it from. So the answer here is very obvious. Now this answer is by inhalation of aerosol in the air-conditioned room. So this Legionnaire's disease, right? So this Legionnaire's disease, this causes atypical or walking or interstitial pneumonia. Atypical or walking or interstitial pneumonia is caused by Legionella pneumophila, right? It also causes, Legionella will also cause Pontiac fever. So this another disease entity, Pontiac fever, which is also caused by Legionella. So this will cause Pontiac fever and it will also cause this Legionnaire's disease, right? So there is a atypical walking interstitial pneumonia. Now these 2 diseases of Legionella, they are by 2 things. Remember about this. Legionella, first is AC. So wherever there is AC, we talk about air conditioning, we talk about Legionella. Second thing is cruise ships. So wherever we talk about AC and cruise ships, then we talk about Legionella. So from air conditioned sources of infection, we can get Legionella, right? So if the patient, let's suppose that in an ICU there are 10 patients and there's an air conditioner at one end. So the patient who is closer to the air conditioner, he or she will get Legionella infection, right? Similarly, in cruise ships also there is Legionnaire's disease. So these 2 things are very, very important. Air conditioned room at a convention center. This is because that's how Legionella has come in this patient, right? Okay, so I wish you all the best and thank you very much.